Welcome, everyone. Today we'll explore a specific type of protein called receptor tyrosine kinase. This protein plays a major role in cell signaling, and when it goes wrong, it can cause cancer. Throughout this lesson, you will understand how these molecular details help researchers design targeted cancer therapies. In today's lesson, we will explain how receptor tyrosine kinase functions in cell signaling. We will also be able to describe how RTKs become activated and transmit cellular messages. Thirdly, we will analyze how mutations in RTKs lead to uncontrolled cell growth. And lastly, we'll explore targeted therapies that block RTK signaling in cancer. As we go through this lesson, follow along throughout the lesson to complete the worksheet. There will be several pause breaks to allow for you to answer questions and think about each topic as we go along. If you have questions, please feel free to jot them down. I can answer them for you at the end of the lesson. To access this worksheet, you must click the link and make a copy for yourself. Before we talk about receptor tyrosine kinases specifically, let's remember how cells talk to each other. Just like we use and messages, cells use ligands and receptors. So throughout this lesson, you will also hear me use RTK instead of receptor tyrosine kinase, as is a big word, so we're just going to shorten it just for this lesson. So throughout this lesson, you will also hear me talk about key terms. Number one is ligand. Ligand, which is a molecule that acts like a message that is being sent to a specific address. It can be either a hormone, a neurotransmitter, or a growth factor. The receptor is a mailbox or a cell phone on the cell surface that receives the message. Think of it this way. Ligand is the text message, the receptor is the phone, and the signal transduction process is the notification, sound, and response. When the phone gets the message, it sets off a series of events. The user reads it, reacts, and maybe sends one back. Instead of the cell, it looks like a chain of proteins being activated, one after another. Once the receptor receives the signal and passes it along inside the cell through a cascade of molecular switches, often through phosphorylation, where one protein activates the next. This is called signal transduction, and it is how cells translate an outside message into a specific response, like turning on genes for growth or division. Now, I would like for you to turn and talk with your neighbor. You will have 3 minutes to discuss why you think cells need complex systems, instead of just allowing things to enter. Now, as you think about this question, think about this. When your body needs a repair wound, growth factors are released that bind to receptors on nearby skin cells. Those receptors trigger cell division to close the wound. However, if that signal keeps going when it shouldn't, that's how cancer starts. Too much communication, but of the wrong kind. Now we'll take a little bit of a pause just to allow for you guys to get some stuff written down. All right, let's move on. Since we have just learned that cells use receptors to receive signals from the outside world, let's look at one of the most important types of receptors. Receptor tyrosine kinase, or RTK. As stated earlier, you guys will hear me shorten it a lot throughout this presentation. It just makes it easier for you to understand. Now, receptors are a large family of membrane proteins. The term membrane spanning means that part of the protein is on the outside of the cell, another part of the cell is on the inside, and the part of it crosses through the cell membrane. The design lets the receptors serve as a bridge that physically connects what is happening on the outside of the cell to what is happening on the inside of the cell. We can take a look at the structure. So if you guys look at your worksheet on part 2, it's going to ask you to utilize 3 different colors to create a diagram labeling the 3 main parts. So we have the extracellular ligand binding domain, the transmembrane helix, and the intracellular tyrosine kinase. If you look at the presentation, you can see how it looks. I would like you to draw, for example, for yourself. As stated, we can look at the structure, and the first part is called the extracellular domain. This is where the ligand binds. You can think of it like the ear of the cell. It listens for chemical signals like growth factors or hormones. The second part is the transmembrane helix. This is a single stretch of amino acids that act like the cord that tethers the receptor into the cell membrane. However, it doesn't do much signaling itself, but it holds everything in place. The last part is the intracellular tyrosine kinase domain. This is where the real action takes place. Once that ligand binds outside the inside domain, it becomes activated and transfers phosphate groups from ATP to specific tyrosine amino acids on nearby proteins. When you think of RTKs, imagine them as a power button on a computer. When the correct ligand presses the button, the receptor turns on and sends the electricity, or in this case, signals through an internal circuitry of the cell. The phosphates are like the electrical sparks that activate the next set of components. Now that we know what RTKs are and what they look like, let's see how they actually get turned on and start the chain reaction inside the cell. This brings us to our next topic, the activation. So on your worksheet, what you're going to do is you're going to be able to match the description to the steps in the activation process. So listen out for what I say and put the letter next to the number that it's associated with. So, now that we have identified the three main parts of the receptor tyrosine kinase, let's see what really happens when the receptor actually becomes activated. Step 1. It begins with ligand, which is often a growth factor or hormone. When that ligand binds to the extracellular domain of the receptor, it changes the receptor's shape. This is structural change is the key to activation. It's like turning a key in a lock. After binding, two receptor molecules move together and pair up. This process is called dimerization. This is one of the most important events in RTK activation. You can think of each receptor like a half of a glow stick. Alone, they can't glow, but when you snap them together, the energy is released and the signal lights up. Once paired up, the receptors activate each other's kinase domains on the inside of the cell. Each kinase transfers phosphate group onto specific tyrosine residues of the other receptor. This is why it's called autophosphorylation. The phosphate groups act as a molecular tag signaling to other proteins that the receptor is active. In the last step, which is after the autophosphorylation, the phosphorylated tyrosine serves as the docking site for specific intracellular signaling proteins. These have regions called SH2 domains that recognize and attach to phosphorylated tyrosines. Once they bind, these proteins become activated and start signaling cascades inside the cell, such as the RAS MAPK pathway or the PI3K AKT pathway. If you're having trouble understanding, think of a receptor as a power strip. When the receptor gets activated, these downstream proteins can plug into it. The phosphate tags are the outlets that allow these signals molecules to connect and get powered up. Now that we see how RTKs activate and transmit signals through phosphorylation, we will move to what happens next. This is where phosphorylation cascade amplifies the message inside the cell. All right. So we are on part 4 of our worksheet. If you guys don't know where we are. So for the thing, what you guys are going to do is you're going to give me a list of how it works and filling in the blanks. Then after that, I will take a break and give you a time to answer the stop and think question, which is what is one advantage of using a phosphorylated cascade in signaling? So let's talk about how it goes from a signal to a response. So I've just shown you guys how RTKs become activated through dimerization and autophosphorylation. So what happens next? The phosphate groups we saw aren't just decorations, they are molecular beacons that attract signaling proteins inside the cell. This begins what was called a phosphorylation cascade. A phosphorylation cascade is just like a row of dominoes. Once you knock one down, the motion continues to trigger a chain reaction. In a cell, each protein phosphorylates and activates the next passing the signal forward until it reaches the nucleus. An example of this is using the best known pathway, which is the RAS MAPK pathway. For this pathway, the activated RTK recruits a small GTP binding protein called RAS. RAS switches from an off state to an on state. The active RAS then activates a protein kinase called RAF or RAF. RAF phosphorylates to MEK, which phosphorylates to ERK. Lastly, ERK travels into the nucleus and turns on genes that control cell division and growth. This process is how cells respond to growth signals. It is normally very controlled and turned on only when needed. In this case of cancer, mutations can make one of these proteins like RAS or RAF permanently active so that the signal never stops. That's like light switches that are jammed in one position. Phosphorylation cascades are powerful because of amplification. Once a receptor can trigger hundreds of RAS molecules, each of which activates hundreds of kinase downstreams. This means a single ligand binding event can cause a massive coordinated cellular response. But with great power comes the need for control. So how do these cells keep these cascades from running wild? That's what we'll take a look at next. The regulatory mechanism that turns RTK signaling off when the job is done. Take a second, look at your papers, and hit the stop and think question. You can always go back into the presentation if you need to. So let's keep it in check. So on this journey, we have seen that RTK being activated to the phosphorylated cascade. Now that we can see how the cell keeps this powerful process under control. If these pathways never turn off, cells could grow indefinitely, leading to diseases like cancer. In science, balance is everything. Receptor tyrosine kinases need to activate quickly when the body needs them, but they also need to deactivate just as efficiently once a signal has been received. Regulation ensures that cells respond only when necessary. Phosphatases, known as the off switch, are enzymes that remove phosphate groups from proteins. Remember, phosphorylation turns proteins on, so the opposite effect, which is dephosphorylation, turns them off. Think of phosphatases as the cleanup crew that comes in after the work is done. They reverse the phosphorylation mark, stopping the cascade and allowing the cell to rest. The second mechanism is receptor internalization. Once the RTK has done its job, it's pulled into the cell by a process called endocytosis. Inside the cell, the receptor can either be recycled back to the surface for reuse or degraded by enzymes. This prevents the receptor from being constantly active at the cell surface. If you would have to compare this, it would be temporarily unplugging a phone that has been ringing nonstop. You remove it from the surface so it can keep receiving calls. The third mechanism is negative feedback loops. The cell uses feedback inhibition to self-regulate. Some of the proteins activated downstream in the cascade, like ERK, which can travel back and inhibit earlier steps in the pathway. This creates a feedback loop that prevents overactivation, kind of like an automatic braking system in a car. So, cells normally keep RTKs under tight control, but in cancer, these safety systems fail. Mutations or overexpression cause the RTK to be permanently on. So on the next slide, we'll see how that looks in real cancer, like lung or breast cancer, and how scientists target these malfunctioning receptors with specific drugs. Now that we have learned how cells normally regulate RTK signaling, let's see what happens when the regulation fails and how it leads directly to cancer. In normal cells, RTKs are like light switches. They turn on briefly to send a signal, then turn off again. However, in many cancers, the switch gets jammed in the on position. This means the receptor keeps telling the cell to grow, divide, and survive, even when no external signal is present. The three main ways this can happen is first through the gain of function mutation. This is a change in the receptor's DNA sequence, and it alters its shape, allowing it to stay active without a ligand. The second mechanism is gene amplification. This is just where the cell produces too many copies of the RTK gene. This leads to overexpression of receptors. More receptors equals stronger signals. The last mechanism is autocrine signaling. This is where some tumor cells actually produce their own ligand. It's like leaving yourself endless voicemail. The signal never stops coming. When errors happen, signaling pathways like RAS, MAPK, and PI3K AKT stays permanently active. The cell continuously divides, avoids death, and may even recruit blood vessels to feed the growing tumor. This uncontrolled signaling is a hallmark of cancer. Now, we have what we call EGFR, which stands for epidermal growth factor receptor. It's one of the most studied RTKs because of its crucial role in regulating how cells grow, divide, and heal after injury. In a healthy cell, the epidermal growth factor activates only when the epidermal growth factor binds to it. This binding triggers dimerization, autophosphorylation, and then cell division. But in certain lung cancers, particularly non-small cell lung cancer, mutations in the EGFR gene causes the receptor to remain active all that time, even without the growth factor present. The mutation usually occurs in the kinase domain, which is part of the receptor that binds ATP and transfers phosphate groups. The mutation reshapes the domain so that it stays locked open, sending continuous signals through the RAS-MAPK and PI3K-AKT pathway. Now we can use this knowledge to design targeted drugs, such as giftonib and erlotinib, which are small molecule inhibitors that compete with ATP for the binding site in EGFR. If ATP cannot bind, phosphorylation cannot happen. This means that the receptor cannot activate downstream signaling. Now that we have seen how the epidermal growth factor receptor drives cancer and how we can target it, we'll move on to another major RTK in cancer, which is known as HER2, and how monoclonal antibodies revolutionize treatment for breast cancer. So, if you guys didn't know anything about breast cancer, HER2 is a human epidermal growth factor receptor number 2. It is one of the 4 closely related RTKs in the ERBB family. In healthy cells, HER2 helps to regulate growth and repair, particularly in breast tissue. And 1 in 5 breast cancer patients, the HER2 gene is amplified, which means the cell has many extra copies of that gene. This leads to overexpression of HER2 receptors on the cell surface. If we imagine a cell with 10 times more growth switches than normal, even with ligands, the HER2 receptor from dimers and can send powerful continuous signals to the nucleus, telling the cells to divide. The constant signaling activates pathways like RAS and PI3K, promoting uncontrolled cell growth and preventing programmed cell death. This is what drives tumor formation in HER2-positive breast cancer. In the late 1990s, scientists developed trastuzumab, which is better known as Herceptin, which is a monoclonal antibody that specifically targets HER2-positive cancer cells. Herceptin binds to the extracellular domain of the HER2 receptor, blocking it from pairing with other receptors. Without dimerization, HER2 can't send growth signals. Not only does Herceptin block the signal, but it also recruits immune cells to attack the HER2-positive tumor cell, which is a process known as antibody-dependent cellular cytotoxicity. This is a dual mechanism that makes Herceptin one of the most successful examples of targeted cancer therapy. So, if you were on your paper and following along as you go, you will see that we have part 7, which deals with a case study. So, what you're going to do is fill out the normal function, what the mutation effect is, and what the drug therapy is to help target the specific issues with lung cancer and breast cancer. Then you are going to answer the stop and think question, which is what makes targeted therapy like Herceptin more precise than chemotherapy. Take a few minutes, and then we're going to move on to part 8, which is the therapeutic approach. Before we do that, we are going to have a turn and talk. So you are going to turn and talk with your neighbor. Alright, so what you're going to do is you're going to turn and talk with your neighbor. And you're going to answer the following question. What is the main difference between Herceptin and small molecule inhibitors such as giftonib? So just know that both the epidermal growth factor receptor and HER2 are RTKs that when overactive they drive cancer, but they have learned to design therapies to specifically target them. So on the next slide, we're going to look at the therapeutic approaches as stated previously. Welcome everyone. Today we'll explore a specific type of protein called receptor tyrosine kinase. This
We have
protein plays
seen
a
how
major role
targeted
in
therapies
cell
like
signaling,
Receptor and
and when it
Giftanim
goes
can be
wrong, it can cause
incredibly
cancer.
ineffective.
Throughout this
But
lesson, you will
unfortunately, they're not
understand how
perfect. So let's
these
explore
molecular
the major
details
types of
help
treatment and
researchers
the
design
challenges
targeted cancer
scientists
therapies.
face when using them. So we have small
In
molecule
today's
inhibitors,
lesson, we will
which are
explain
drugs
how
that are small enough
receptor
to enter the
tyrosine
cell and
kinase
block the ATP
functions
binding
in
site of
cell signaling.
RTKs.
We will also
Without
be able to
ATP, the
describe how
receptor can
RTKs
phosphorylate
become
itself or other
activated
proteins.
and
Some
transmit
examples
cellular
include the
messages.
giftanin
Thirdly, we
and
will analyze
or
how
lotimin
much
for the
how
epidermal growth
mutations in
factor
RTKs
and
lead to uncontrolled cell growth. And
melatonin
lastly,
for
we'll
HER2.
explore
The
targeted
advantage
therapies
is that
that
they
block
directly target
RTK
the source of
signaling in cancer.
overactivity, but
As
cancer
we
cells
go through this
often
lesson,
mutate
follow
the
along throughout
binding
the lesson to
site,
complete the
making the
worksheet.
drug
There
ineffective
will be
over
several
time.
pause breaks to allow for you to answer
Monoclonal
questions and
antibodies
think
target the
about each
receptor
topic as we go along.
extracellular
If you
domain
have
to
questions,
prevent ligand
please feel
binding
free to
or
jot them down.
dimerization.
I can answer
Due to the
them for
fact
you at
that
the end of the
antibodies
lesson.
act outside the cell, they are highly
To
specific,
access
but they're
this
also large molecules that
worksheet, you
must be
must click
given
the link and make a
intravenously
copy for
and
yourself.
can't reach receptors deep inside the body easily. A
Before we
combination
talk
of
about
therapy is
receptor
great as
tyrosine
well.
kinases,
This is where doctors
specifically, let's
often use a
remember how
combination
cells
of
talk
drugs
to
that
each other.
target multiple
Just
points
like we
in the
use
pathway.
phones and
For
messages,
example,
cells use
combining
ligands and
a
receptors.
HER2 inhibitor with
So
a
throughout this lesson, you will also hear
PI3K
me
inhibitor
use
helps to prevent
RTK
cancer cells from
instead
switching
of
to backup
receptor
pathways. This strategy is like
tyrosine kinase,
blocking
as
multiple
it's a big
exits in a
word, so
maze,
we're just going to shorten
making it
it just
harder
for
for
this
cancer
lesson.
cells to escape.
So throughout this lesson, you'll
Even with
also
targeted
hear me
therapy,
talk about
cancers
key
can be
terms.
tricky.
Number
Resistant
one is
development,
ligand. Ligand, which is a
cancer cells
molecule that acts like a
mutate
message
or
that is being
activate
sent to a specific
alternate
address.
pathways.
It can be either a hormone, a
Side effects
neurotransmitter, or
happen
a growth
because
factor. The
RTKs also
receptor
play a
is a
role in
mailbox or
normal
a cell
cells,
phone on the
especially in the
cell
skin,
surface
gut, and
that
heart.
receives the message. Think of
Tumoral
it this way.
heterogeneity
Ligand
means
is
not
the
all
text
cells in the
message,
tumor have the
the
same
receptor is the
mutation,
phone, and
which
the
makes
signal
it harder to
transduction
eliminate
process
all of them.
is the
The future of
notification
RTK
sound
therapies
and
lies in
response.
personalized
When the
medicine,
phone gets
analyzing
the
a patient's
message, it
tumor
sets off a
genome
series
to
of
select
events.
the most
The
effective
user reads
drug.
it, reacts, and maybe sends one back.
So, we have now seen how traditional inhibitors and antibodies revolutionize treatment, but cancer continues to adapt. The next frontier is personalized medicine. This is where treatment is tailored to each patient's genetic. Every cancer is genetically unique. Using next generation sequencing, oncologists can identify exactly which RTK or pathway is mutated. This lets them select a targeted therapy rather than relying on broad chemotherapy. Through research, there is a development of new types of treatments. The first type of treatment is called biospecific antibodies. This is where they can bind two different receptors such as HER2 and HER3, blocking crosstalk between the pathways. The second is antibody drug conjugates, which are like guided missiles. Herceptin can be attached to a chemotherapy drug to deliver it only to HER2 positive cells. RTK degraders don't just block receptors, they tag them for destruction by the proteasome. All right, we should be on part 9 of our papers. If you have any questions, let me know. Now, let's have some key takeaways. So, let's pull everything together to make sure that we understand what we have been talking about this whole time, because we've been talking about a lot, and I want to make sure that you guys understand. So, RTKs are what we call a communication hub. They control how cells grow and respond to their environment. When the system works, it works great. However, it works great due to the fact that precise and self-limiting happens. But when it fails, it can drive cancer. Activation happens when binding triggers dimerization and phosphorylation, launching cascades like MAPK and PI3K. Mutations or gene amplification in EGFR and HER2 turn these receptors into permanently active switches during uncontrolled cell division. Targeted therapies such as Giftanim and Herceptin show how understanding molecular mechanisms lead directly to life-saving treatments. What do you think the biggest challenge is, though? Is it detecting the right mutation, designing the drug, or preventing resistance? Tell me why. And now that you guys have mastered the science, it is your job to apply what you have learned and complete your worksheet. So let's make sure that we have everything written down. So in your worksheet, you will be having a cell communication refresher where you are going to tell me what cell signaling is in your own words. Tell me what a ligand is and give me an example of a ligand. You can tell me what is a receptor and where it is located.
After you answer that, there's some questions for you to answer to stop and think about. The next part is having the structure of the receptor tyrosine kinase. That picture has been shown to you, and you guys can utilize that picture to draw it on your thing and label it out and color it. Coloring it and labeling is a really good strategy for taking a test. Now, we also have where you give me the function of the parts of the cell and for you to give me an analogy that was given from this lesson. Listen specifically to the specific parts that are a part of the extracellular domain or the transmembrane helix, and that is where you can find the analogy to this particular part. Now, as stated for part 3, this is where you guys are going to match the letter to the number, and you guys are going to place it right beside it. Part 4 has you filling in the blanks and explaining the process as it relates to how the phosphorylation cascade happens. And then you have a critically thought question. I want to know, why could a mutation in RAS or RAF lead to cancer even if the receptor is normal? If we go to part 5, that's where you give me a description of the regulations of RTK signaling and give me an analogy. Those analogies will help you understand on the test so that when you see it again, that you can have a basic understanding of what they'll mean. Also, in the worksheet, you're going to give me the mechanism and the result of that mechanism as it relates to mutation being overexpressed. I talked about part 7, where you guys give me the normal function of HER2 as it relates to breast cancer and EGFR as it relates to lung cancer. You'll also give me the mutation effect in the drug therapy that is a part of it. Now, we also want to talk about the therapy type. Give me an example, the main target, and the challenge that is associated with it, because we want to make sure that you understand that. Lastly, you're going to fill in the blank, and part 9 and part 10 is a reflection. You will also have an exit ticket. Please make sure that you turn it in at the end of class. And thank you for watching my presentation on receptor tyrosine kinase. I am Kennedy Dorsey, and this is my presentation.